Hello, I'm Jasmine Sweeney, and I was assigned unknown number 6, which I later found out to be a gram-positive Bacillus megaterium and a gram-negative Enterobacter cloacae. My gram-positive Bacillus megaterium, its natural habitat is mainly in soil, but it's also found in other places like honey, milk, seawater, etc. On my slide, I put the taxonomic hierarchy for Bacillus megaterium. A little background is that it's a gram-positive rod-shaped organism. It is spore-forming, so unlike your typical gram-positive, which are easy to kill, Bacillus megaterium is actually more difficult. Effective way to kill Bacillus megaterium is with autoclave. It kills the spores and denatures the proteins in them. Bacillus megaterium is big, hence the mega in megaterium. It's rarely associated with pathogens. It also produces a antiviral agent. Bacillus megaterium isn't associated with any major diseases. Bacillus anthracis, on the other hand, is known to cause a serious infection known as anthrax. There are 4 different types of infections associated with anthrax. You could get it cutaneously, which is when small blisters or bumps form on the skin. After that, you'll see a painless skin ulcer with a black center. You could also get it by inhalation and gastrointestinal, which will cause fever, chills, headaches, nausea, etc. You could get it by injection. And if you get it this route, you will have a deep abscess beneath the skin where the drug was injected. There are different treatment options for anthrax. They'll typically prescribe antibiotics, also known as doxycycline, or they will give you an antitoxin. For more severe cases, of course, you can be hospitalized where they will actually have to drain the fluid. Prevention of anthrax is from a vaccine. They have a AVA vaccine. Antibiotics are also prescribed to prevent the development of anthrax if you've been exposed to it. Anthrax is associated with 3 virulence factors. They're known as DPGA, LT, and ET. ET and LT are exotoxins, and DPGA is a capsule. For my scientific article, I did it on Bacillus megaterium strain 1259. It was used as a probiotic to examine its effects it had on lactating dairy cows' performance. So what they did is they got Bacillus megaterium 1259 and isolated it from chicken manure, kind of gross. They used 24 lactating Holstein dairy cows. They randomly assigned them to 4 groups, so 6 cows in each groups. They did have 1 control group. So on the bottom here, you see control T1, T2, T3. The control group, they were fed the same TMR diet, which is a mixed diet. The only difference with the control group is that it didn't contain the probiotic, but the others did. As you can see here, it was significantly higher in T3 for 4% fat corrected milk, which pretty much is the energy in the milk. So they produced more, which was the highest dosage they gave to that group of cows at 15 grams. So at the end, they came to the conclusion that Bacillus megaterium strain 1259 is potentially useful as a probiotic in dairy cows. For my gram negative, I got Enterobacter cloacae. Its natural habitat is found in terrestrial, aquatic, and the intestinal tracts of humans and animals. I posted its taxonomic hierarchy right here. A little background on Enterobacter cloacae is that it's a gram-negative organism, which makes it difficult to kill considering it has that lipopolysaccharide outer membrane. It's rod-shaped. It's motile. It has paratricious flagella, which means it has flagella all around it. Its optimum temperature is 37 degrees. Enterobacter cloacae is associated with nosocomial infections, which is an infection or toxin that exists in a certain location, such as in this case, a hospital. The type of infections it causes is ammonia, urinary tract, wound infections, and meningitis. I'm going to focus on urinary tract infections. Treatment for this is nitrofurantoin. It's a synthetic drug used to treat bladder infections because it concentrates in the urine and the kidneys remove it from the blood. It's known as a protein synthesis inhibitor. Prevention of UTIs are drinking lots of water for women, wiping from front to back. E. cloacae isn't associated with any virulence factors, but Escherichia coli is. E. coli is associated with adhesions, aerobactin system, hemolysin, capsules, and resistance to serum killing. So for my scientific article for my gram negative, I did it on Forsythia suspensa polysaccharide, known as FSP, and its effects that it has on Enterobacter cloacae. Enterobacter cloacae contains an enzyme known as SHV12, extended spectrum beta lactamases. Pretty much what that means is that it has the ability to cut the beta lactam ring in penicillins and synthetic penicillins. So what they found out from the study was that this plant had the ability to suppress the enzyme that Enterobacter cloacae produces, which is huge because this enzyme is what makes it resistant to antibiotics, which is a big problem we're dealing with because soon every single bacteria will be resistant to antibiotics. So finding out that FSP is able to inhibit the growth, I think it's amazing. And this is my references. Thanks for listening. Bye.